Colleagues, good afternoon from Manchester. We are going to start the meeting now. So hello everyone and welcome back to the second day. Without further ado, I'd like to introduce our first speaker, Emily Duderstadt from the University of Louisville. Emily.
So in this talk, I'll be presenting our congenic mapping and phenotyping of mammary carcinoma susceptibility locus 3 in the rat genome, and then some comparative analysis in the human orthologous regions. So you can see from this ideogram here that the rat genome contains multiple QTLs that modulate mammary cancer risk, but our focus here is on MCS3 on rat chromosome. So rat MCS3 was previously mapped to 29.4 megabases on rat chromosome 1 using Worcester Copenhagen Congenics. You can see here in the tumor multiplicity table that the Worcester strain is highly susceptible to chemical induction of mammary cancer and develops numerous tumors per rat, while the Copenhagen strain is resistant and virtually develops no tumors. And so introgression of differing portions on the right here of the Copenhagen strain of the predicted Copenhagen allele onto a Worcester susceptible background resulted in congenic lines with different tumor multiplicity phenotypes seen here in the table. And so line A and D showed a resistance phenotype, while lines E and G showed a susceptibility phenotype. And then using the overlap of resistant line D with susceptible line E allowed MCS3 to originally be narrowed to 29 megabases. And the Copenhagen allele here caused around a 50% reduction in mammary cancer susceptibility. Now, in order to further map and delimit this locus using Worcester Copenhagen Congenics, I needed to create new congenic lines. So I began here with MCS3 line D, which was a resistant line. And I crossed with the parental Worcester susceptible strain to get hets. And these hets I then back crossed to the Worcester strain again. And then I screened offspring for informative recombination events with shortened MCS3 intervals to use as founders of new congenic lines. So that these I then back crossed to the Worcester strain to expand this genotype, and then did brother-sister matings to then fix these lines. So here's the resulting congenic map of these MCS3 lines that I established. And remember, all of these are on a Worcester susceptible background, except for differing portions of this Copenhagen allele shown in black. And so once congenic lines are created, we have to phenotype them in order to determine if a genetic introgression results in retained mammary cancer susceptibility or resistance. So in our lab, to induce mammary cancer, we administer the chemical carcinogen 7,12-dimethylbenzanthracene, or DMBA, at 50 to 55 days of age in the rat. And this is a peak pubescent time window of mammary gland susceptibility to carcinogenesis. And then we assess tumor multiplicity 15 weeks following administration. So the DMBA model is the most efficient method to study human breast cancer, as tumors develop quickly and in the form of invasive ductal carcinomas, which account for more than 80% of diagnosed female breast cancers. DMBA itself is highly lipophilic and it accumulates in fatty mammary tissue. And so this presents a highly efficacious model then, because DMBA is directly bioactivated in the mammary gland, and then it's here that it acts as a mammary gland specific carcinogen. So here is the tumor data from each of my MCS3 congenic lines. So remember, line D was my original line from which my congenics were derived and showed a resistance phenotype of around 3 mammary tumors per rat. And then my Worcester susceptible control showed a high tumor multiplicity count of around 8 mammary tumors per rat. And congenic lines H, I, and K showed similarly high mammary tumor counts, but line J showed a resistance phenotype similar to line D of around 3 mammary tumors per rat. And our post-hoc analysis compared each of these congenics to the Worcester susceptible control, and only line J had a significantly lower tumor distribution. And so now we can add these phenotypes onto the congenic map here on the right, and we can use the overlap of my susceptible line I with resistant line J to then narrow this region by 75% to only 7.2 megabase region. So I also took weights at the time of tumor multiplicity counts 15 weeks following DMBA administration. And I found that line J, which you remember was our only resistant line, also had a lower body mass compared to the other congenics. And so I wanted to investigate this phenotype further. So then I started taking weights from our regularly housed breeders over different developmental time points to look at growth. And so here I compared Worcester versus line J, looking at both male and female rats. And I found here that MCS3 genotype also affected male body mass, as line J males, shown in light blue here, were significantly smaller than their Wüstefurth counterparts in dark blue. But at this point, I didn't see an effect on female body mass. So then I next moved line J and Wüstefurth females into all female housing, which is what we do for rats that are going to be given DMBA. And it's here now that I found a similar phenotype where line J females were significantly smaller than their Wüstefurth counterparts. Again, the only difference here being the lack of male rats in the room. And note that I do have differences in scale here from these independent experiments. And so at this point, we've now phenotyped rat MCS3, which we found had pleiotropic effects on both mammary cancer susceptibility as well as body mass. And now we could move on to comparative genomics. And so here is the original 29 megabase rat MCS3 region on the right, and the human orthologous synthetic regions to it on the left. And then with my Congenics, I narrowed MCS3 to only the 7.2 megabase region, and this reduced the pool of nominated genes from 310 to 44 genes in this rat region. And now we can focus on these human synthetic regions here for comparative analysis. So in doing so, we found that there are 23 genes in common between rat MCS3 and the orthologous human regions. And there are an additional 12 genes in the human synthetics that are not annotated in the rat genome. And then additionally, there are 17 uncharacterized long non-coding RNAs as well as 4 microRNAs. So now our MCS3 rat model can be used to evaluate potentially overlapping breast and human. We found that human 15q 25.1 to 25.2 synthetic region to rat MCS3 is most relevant to our studies. It contains this one breast cancer risk variant, which may be marking this entire allele as a true positive association with female breast cancer. And also in the same region, there are 67 variants that are associated with body mass GWAS traits, such as body mass index, body fat distribution, and body fat percentage. And so now human and rat comparative genomics is now informative for candidate genes to focus on. And this gives us rationale here to focus on this region in the rat for mechanistic analysis of this human allele. And so of the 17 genes in this priority region, there are 7 that I listed here on the right that are previously associated with female breast cancer. And so now these nominated orthologous genes will need to be evaluated in terms of disease susceptibility. So here again are those 7 genes in that priority rat MCS3 region that are correlated with human breast cancer already. So TM6SF1 is a transmembrane protein whose promoter is differentially methylated in malignant breast tissue. c-Myc positively regulates epithelial to mesenchymal transition and has elevated protein levels in tumor tissue. IL-16 is a pleiotropic cytokine. It's elevated at both the RNA and protein level in patient serum, spheroids, as well as tumor xenograft models. EFL1 activates ribosome and is found to be differentially expressed in tumor versus non-disease breast tissue. BMC1 has a proliferative function, and it's differentially methylated in metastatic breast cancer tissue as compared to primary. ARNT2 is a transcription factor that responds to both developmental as well as environmental stimuli, and it's been found that there's elevated expression in tumor versus non-disease breast tissue. And then finally, ABHD17C depalmitoylates the proto-oncogene NRAS, and its expression is elevated in patient-derived tumor xenograft models. And one specific interesting gene to note here is ADAM-TS-like3 gene, which is nominated from both rat MCS3 as well as human 15q 25.2, because it houses 55% of those 67 GWAS body mass variants mapped to this region. And additionally, it's located nearby to this breast cancer risk-associated variant, which I already said may be nominating this entire allele as associated with true breast cancer risk. And looking further, with Oncomine data, its overexpression nearly 4-fold in invasive breast tumors as compared to non-disease breast tissue. And then it's also found to be highly overexpressed in epithelial alveolar cells here, which is a major cell type of the mammary gland. And so its exact function remains elusive, but it does potentially have a role in cell matrix interactions or assembly of extracellular matrices. So in summary, I narrowed MCS3 to 7.2 megabases, and it contains orthologous human synthetic regions. And And so the priority here is to utilize comparative genomics of human 15q 25.1 to 25.2. But if this proves to not be informative, then line J can be used to collect additional recombinants and create new congenic lines in order to further narrow this locus. Also found that MCS3 has a pleiotropic effect on mammary tumor susceptibility as well as body mass. So MCS3 susceptibility genes may also be functioning through growth and development. The effect of MCS3 genotype on female body mass was only seen in the absence of males. So this suggests that male presence may be having a pheromone, visual, or auditory effect on the MCS3. And finally, the MCS3 orthologous human 15q 25.1 to 25.2 is associated with breast cancer risk as well as body mass. And so this gives rationale to ultrafine map the human region for breast cancer risk, as well as to identify the MCS3 causal genes. And with that, I can take any questions.
That's great, Emily. Super. Thanks very much for this fascinating talk. I think your microphone might have been just by your keyboard because there was background noise there. Maybe that was the reason why sometimes you couldn't quite hear so well. But thanks very much for this talk. Do we have any questions for Emily? You can use either your virtual hand or put a question in the chat, or your real hand. Abe, you've got a real hand up.
Yeah. Emily, in the congenic region, I noticed that there were some CYP genes, which are P450 enzymes. And I wondered if you know how that chemical that you're using to induce the tumors is metabolized, and if it's possible that those are the genes that are responsible for clearing it, in which case the susceptibility could be differential metabolism of this carcinogen.
Yeah, so I am a little bit rusty on that. It's metabolized by CYP1B1, if I remember. So it's not these specific.
So not the ones there.
Yeah.
Is it easy to measure this thing? I mean, I wonder if you could measure after injection to see if the two congenic and wild type are clearing it equally. I don't know how feasible it is to make a measurement like that, but that would, right?
Yeah, I'm not sure. I'm not sure if that's ever been attempted or not.
Yeah. Okay. Good. Thanks.
Mm-hmm.
Emily, could I ask you a question? Your third point on your last slide here, that the male presence has an effect, or the effect of MSC3 is only seen when the males are absent. So I just wonder sort of what's the functional aspect of that is, how could this possibly work? That seems to be quite an unusual observation.
Yeah, and that's what we're struggling with as well. We're not really sure what to make of this because male rats are never in the cage. It's just their presence in the room. So it has to be something, either an auditory or visual thing that's going on, or something at the pheromone level traveling through the ambient air. But this is still very preliminary, and we're not sure what to make of it just yet.
So they're not, are they kept in IVCs, so in individually ventilated cages, or are they in open cages?
It's an open system.
Oh, right, okay.
But they're not in this in the same house.
Very interesting. All right, thanks. Yeah, you've got a couple of questions in the chat if you wish to look at them, but we're going to move on to our next speaker. And our next speaker is going to be Manal Taba from the University of Southern California.
And the whole genome sequencing was derived from a spleen from the founders. We got high coverage paired end reads, 150 base pairs from one sample per substrain. And the RNA-seq was derived from hippocampus, as I mentioned, the data is 50 base pair single end reads from 6 to 11 male samples per substrain. The whole genome sequencing data was analyzed by calling variants, SNPs and indels, structural variants with two different softwares, lumpy and c-eminators, and merged the structural variants. The gene dosage in or the segmental duplication dosage was inferred from depth signal in sec-dupe annotations, and STR was also were also called by Hipster. RNA-seq data was normalized, and we found about 300 DE genes among these substrains. And following that, we did association studies with linear mixed models using genomic relatedness matrix to control for relatedness in these samples. And significant associations was inferred by FDR corrections. So the first observation, we looked at the distribution of different variants in these call sets, and we found variation hotspots present throughout the genome in certain segments. And all of these variations were clustered in the same or similar regions around the genome. You can see chromosome 4, 8, 11, and so on. So we asked, what is the significance of these regions? So if we segregate in each variation type among different substrains, we saw that these variations actually come from B10 substrains, and all of them are common between these 5 B10 substrains, and they consist of 28 segments, which comprise only 5% of the genome and common in B10 substrains. And interestingly, about 90% of total SNPs was actually B10 specific. So the question was, where do these segments come from? So next, we looked at the SNP data from MGI and compared our B10 specific SNPs to them. And the data set from MGI has about 88 substrains from domesticus and musculus origin. So we required at least 300 common SNPs in each of these segments, and more than 90% genotype concordance. And here on the image in the y-axis, you see 24 segments past this requirement, and each segment have similarity to some substrain from MGI database, and we can see that longer segments, we saw fewer matches among the database. And this may infer that these regions are integrated from a newer. So the integration that we hypothesize that these SNPs are from an integration process during the breeding stages, and the larger, the longer segments can come from a newer event, and the older, the smaller ones can, which are common between more substrains, they can come from an older event. So this was the hypothesis that these segments coming from some errors or some problems in the breeding stages and integrate into the genome of black 10. The next, we looked at association between expression data and variations that we found. So genomic variation was tested was loss of function, SNPs and indels, gene intersecting SVs, and gene intersecting sec-dupes and STRs. And as I mentioned, we use LMM, and the FDR correction was done with Benjamini-Hochberg method. And we found 53 significant genes, and they are, as you see in the QQ plot, they are very much enriched in sec-dupe regions, and a dozen of SVs and indels was also significant. And as you can see, we compared these associations with missense SNPs and indels, and they or intergenic SVs and STR, and they actually fall onto the straight line. So about the causality of these associations, generally we can claim causality. However, in some cases, we have a strong prior for causality. For example, in this gene, in KCC2, we found 6J-EIJ has a lower expression than all the other substrains. And among the significant cases or associations, there is one indel, which is a frame shift loss of function. So this particular variation has a larger probability to be causal. This is one case of that we can infer some causality in these associations. Another mechanism for these associations is gene dosage in sec-dupe regions, and as I mentioned, this signal is the strongest in our QQ plot, and most number of significant associations are these cases. And the mechanism is that in sec-dupe regions, the genes have different dosages in different substrains. And as you can see, the number of copy numbers associate with the expression in these substrains. Another interesting case that we found was a duplication in 3 exons in WDFY1 gene. So the middle 3 exons is duplicated in 6J and also present in the reference. And this is associated with expression reduction in 6J. We asked if the additional exons introduced in the middle of the gene can it cause nonsense mediated decay. So to address this, we compared nonsense NMD impaired samples or embryonic stem cells expression data from wild type data. In the next slide. If we look at the sashimi plots for these samples, so the top one is BOMTAC, which doesn't have any junctions between exon 4B and 6A because it doesn't have the duplication. The black 6 has some junctions here. The wild type in the embryonic stem cell expression data has some junctions, but the knockout sample has a lot more junctions. And first of all, we can see that in the bottom right figure, we can see that the knockout expression for WDFY1 is significantly larger than the wild type. And also, if we look at the normalized number of junctions between these two exons, we see that the knockout has significantly larger number of junctions between these two. So we can infer nonsense mediated decay is likely the mechanism for decreasing the expression in the wild type sample. Black 6J. So in conclusion, the different genomic variations were found in 14 closely related black 6 and 10 substrains. 28 potentially intergressed regions were found, introduced in black 10 substrains, and associations tests were performed in expression and genomic variations, and 53 gene associations were found with significant association, and a duplication of 6J and reference genome was attributed to nonsense mediated decay mechanism. So the data is available, so the manuscript is available in bioRxiv, the whole genome sequencing FASTQ files is available in SRA, and process data, you can access it in Mendeley database. Wanted to thank Dr. Palmer and Palmer Lab for helping to analyze and helping to process this project. Also, Jonathan Sabat had a significant role in this project. Also, Dr. Jimbrek for STR annotations and STR calling, and Dr. Wilkinson for providing the embryonic stem cell data to us. Thank you. I'll be happy to answer questions.
Thank you very much, Milad. That's great. Thank you. Any questions for Milad?
There's one from Enza. Milad, so if you just scroll through your chat. Enza, if you're there, go ahead and ask it.
Yes, I was trying to unmute. Very nice talk. I really like that. I was just curious about the sites of the segmental duplication that you're able to detect, provided giving the Illumina sequencing, and just curious about it.
You mean the size of the segments?
Yes. What's the average size of segmental duplication? Because they.
Mm-hmm. So the segdup regions are what is already available in the genome, annotated in the genome. I don't have a size distribution off my head, but they generally are kilobases to 100 kilobases can be that long. They're not short. So although they can be in some cases, but they can generally span multiple genes in some cases, or one or a few genes.
All right, thank you.
But as you said, there are smaller segdup regions as well.
Okay, because that's the strongest signal you have, and I was just curious how, if you're completely able to describe the segmental duplication provided the sites, it's quite big. We're just curious about the range. Thank you.
I think this is a question for the whole team, Milad, but I'll ask you since you're the point person today. If you do long read sequencing here, of course, you'll get quite a bit. You'll be able to extract more out of these fabulous samples. And I'm just wondering where you are with that, because this would be wonderful for coalescent theory. You could figure out whether what you call the events are breeding screw-ups that happened in the 1910 to 1925 in Massachusetts at the farm, or whether they were breeding errors that happened at Jack's, or breeding errors that happened in all the vendors. But just, so tell us more if time permits.
Absolutely, it's a great point. I think 14 substrains is really doable for hi-fi sequencing, and it will give us a hugely better view of these regions, first of all, and we can call variants directly from hi-fi read, and we can just we can go through all these regions, for example, segdup region, STR regions. Yeah, and this is a great idea to supplement this data with hi-fi, especially hi-fi sequencing.
I mean, Rob, you might be aware that we've been posting pictures on Twitter of the new sequel 2 that we bought recently with Jonathan. So definitely, we're thinking about what samples might go on to that.
Cool.
Yeah. In terms of what I think about the introgress regions, they're all in the 10s and none of them are in the 6s. And so I think around the time of the split between the 10s and the 6s, and Milad had an early slide showing when that was historically, but about 1920, I think. Either they were segregating those regions and they fixed differentially in the two lines, the two lineages, the B10 and the B6 lineage, or they were introduced into one of the two before they were further subdivided. And remember, they could have really been introduced into B6. They're different, but we can't say what the ancestral one looked like. And the fact that the segments, some were very short, makes you think about the segments that might have still been segregating, weren't fully inbred when 6 and 10 were split. But then those longer ones look a little bit more like a breeding error that is consistent across all the 6s or all the 10s. But
Yeah.
it's hard to say. None of them perfectly match any modern day strain, but that's maybe not surprising. The origin of those segments may not be perfectly represented among the mice we have access to today.
Very
Does
cool.
that make sense?
Yeah. Reading David Reich on intra-African migration last night, and oh my God, it reminds me of what you've got here with the San being chased around by the Bantus and.
Okay, I actually have to be on a different call, so people may want to keep talking to Milad during the break, but I've got to go.
Yeah, we've got a break now, 15 minutes. I mean, feel free, of course, to continue the conversation, but we'll resume the talks again at 1/4 past.
Yeah. So is there any, just in the conversation department, is there anybody here who actually knows coalescent theory? I sure don't, but it seems like this would be a great application. I'd like to hear about it, if that's true or false.
Hey, Rob, it's Gary.
Yeah.
Requires a random mating population assumption that is clearly way not going to hold here. The way these strains are maintained. Just a thought. Good idea, though.
Okay, so you can't track it back, you're saying, Gary, because of the extremely low N?
No, I mean, you can track it back, but I mean, coalescent theory is not going to be helpful because it's all about what happens in a random mating context and, how far back to the most recent common ancestor, But if you have a new sequencer and you can sequence the heck out of these ancient samples, you could certainly, as they've done, figure it all out. It's beautiful. Cool. And yeah, those breeding errors undoubtedly happened to jacks in the 20s and 30s, and they still happen.
But there was Castaneous in there, and Eva didn't bring the Castaneous in until when, the 80s maybe?
Oh, Castaneous has big chunks of C57 black 6 in it. Yes, we know.
Oh, okay.
In case you didn't know.
Jacks, or do you think that happened in Japan or somewhere?
I think it happened in Tennessee.
I like that thought. We're not very good at interbreeding in general.
No, it's great to see people looking at these substrains. They're the thing that gets under my skin sometimes is people think a mouse is a mouse because it has a label, and those labels don't always mean exactly what you think they mean. So it's so easy to sequence a genome today. You just need to sequence everything or be close enough to something that has been sequenced that you actually know what genome you're working with.
Amen. Yeah, Enza, who's on here today, Gary, is constructing a pangenome of the BXD family, kind of as a proof of principle that we have the software stack working, that we can call the variants from within the pangenome workflow rather than do it against reference.
Yeah, I mean, that's a perfect example. A D segment is a D segment is a D segment, right? Unless it's an old one or a new one, or maybe. Yeah. And I think I don't want to dominate the conversation here, but when we went in and sequenced the collaborative cross strains, oh my word! I mean, mutations have really happened, and they fix pretty fast in these really constrained breeding situations. And there's some really fascinating private mutations. In fact, one of the talks earlier today, looking at CC strains, it's probably worthwhile to just go back and look at the de novo mutations in some of those strains and see if you can't get lucky. Maybe, you know, something's in a functionally obvious gene, and you could do the follow-up work and get a home run, just like people do with other substrain analyses.
Yep, cheat when you can. Use those rare variants like Mendel did.
Yeah, you get to the top any way you can, right? Great.
Yeah. Gary, you'll like this story. I don't know how many, please interrupt if anybody else wants to talk real science, but I've been stuck teaching the first year medical students human genetics, and oh my God, it's like throwing yourself in the past at least 40 years. So we teach the M1 students anything but what's current. So I know a hell of a lot more Mendelian genetics than I did a month ago. It's kind of sad.
It's good to have a historical perspective on things, Rob.
Yeah. You know, for those of you who haven't read the original Mendel paper, there's a really good English translation, which I could plunk it in the in the chat box here. But oh my God, that paper is so good. The introduction to Mendel's 1866 paper, that could be a Cell introduction. It's so good. He's very explicit about what kind of biases he's adding, his quantitative approach. It's just a thing of beauty. Let me see if I can find that.
Title slide where I was talking about Michael Gould and Jim Scholl's work to identify, using QTL mapping, these regions in the rat genome that contribute to mammary carcinoma susceptibility. And one of the things I'll add on here is that this is nice because we can actually study resistance in the rat, because the Copenhagen and Worcester-Kyoto strains are resistant to developing these tumors. So when we introgress those alleles into a susceptible background, you can look for the reduction in tumor number. So that's what we see here. So this is the MCS1B locus, Copenhagen alleles introgressed into a Wista Firth. And so the Wista Firth gets around 7 to 8 tumors per rat, whereas these Congenics get 50% less tumors when it's homozygous. So this was originally mapped to 1.8 megabases on chromosome 2 in the rat using a series of congenic lines. So pretty standard approach. Rat MCS1B, we did sequence the open reading frames in that region and didn't find any protein coding variants that would possibly explain the phenotype. We did identify MIR3 in this study as a priority candidate based on mammary gland gene expression of this gene. It was downregulated in the resistant rats, and whether or not DMBA was present or not. And we also saw a pleiotropic effect of 1B on body mass, whereas the rats with the Copenhagen alleles had lower body mass. It's a quantitative difference, so it was slight. Rat MCS1B contains sequence that is homozygous or orthologous to human chromosome 5. And this 5q11 was one of the first breast cancer risk alleles identified in a Human-GWAS study. And it's 250 kb haplotype block is where that associated variant exists. The candidate genes identified or nominated by that study were MAP3K1, SETD9, MIR3, and this allele also has a slight effect on female stature. All right, so the 3 candidate genes nominated by the Human-GWAS study are MAP3K1, SETD9, and MIR3. MAP3K1 is a known breast cancer driver gene. It's mutated, somatic mutations occur in about 15% of breast cancers. Majority of these are truncating mutations, meaning that it activates the protein. And also higher expression is associated with increased survival. So this MAP3K1 looks like more of a tumor suppressor. The actual functions are known. It's thought to work through the JNK kinase, but this disagrees with our rat data where we saw lower expression of MAP3K in our MCS1B congenics that were resistant. But so definitely a breast cancer gene, but may not be responsible for the susceptibility phenotype we're seeing. SETD9 is also in the human, but it's not conserved between humans and mice or rats through 5' and 3' UTR degeneration. The mice or rats don't have those sequences conserved. So we don't see expression of the rat ortholog in mammary glands from the rat. MIR3, again, this is the focus of this talk today, this gene, and it has been seen to be increased in invasive ductal breast carcinoma compared to non-disease tissue. You can see that here. These are data from Oncomine, where you can go in and check your favorite gene and see if it's differentially expressed. So it gives you some, at least good feelings that you're on the right track when you can see something like this. But it's not the end of the story. There's also a recent study that came out where this group, Hong et al., studied MIR3 expression in the standard approach that you would do in cell culture. So they manipulated expression, they knocked it down, overexpressed it in cancer cell lines, and then measured cancer-related phenotypes like scratch assays and growth and all those pointed to a MIR3 being an oncogene from their studies. Okay. So to move that project forward in the rat, we hadn't seen any interesting sequence variants in open reading frames, so we went ahead and sequenced that 1.8 megabases, the alleles in the Copenhagen strain and the Worcester first strain. And you can see we did this a while ago. Probably a lot of people on this talk don't even know what 454 sequencing is, or maybe they've heard of it. But we did use targeted sequence capture using Nimblegen custom arrays. We got about 20-fold enrichment, and then the sequencing, you can see the coverage we got there. So we covered that just short of 2 megabases pretty well, we think. We missed some repetitive regions and things like that. And at the same time, another group was studying that human locus, and they took publicly available variants, and then they did a fine mapping with population-based cases and controls for breast cancer, and they tested or imputed about 909 genotypes, is what they say in their paper, and about just under 200 of those were had genome-wide significance for breast cancer risk. And they were divided into 3 correlated SNP bins. So we can look at what that looked like here. So this is taking their study in the Santa Cruz genome browser and overlaying their SNPs from their paper. And you can see the 193 variants here, and then they're showing they have these 3 different bins that are independently associated. And you can see the transcripts here. But the real interesting here, these are our results from sequencing the rat. This is the human haplotype block orthologous region here. So we resequenced all this and the flanking regions. Most of the variants we found between the 2 strains were here on the edges. There's quite a few of those. And then within that shared region of synteny, between humans and rats, we found 3 single nucleotide variants. So these would be our candidate variants. We have studied those a little bit in cell culture, putting them in plasmid vectors, and I won't show you those data, but they do, one of these SNPs does recapitulate what we see in expression, where the Copenhagen variant has lower reporter activity than the Wissenfurth variant. Okay, so that earlier work on expression was done in whole mammary gland tissue, and the site of action in breast cancer and rat mammary cancer is believed to be in cells that line the duct. So we wanted to know something about mammary epithelial cells. We did this quick approach where you can enzymatically digest whole mammary glands, and then you get a mammary epithelial cell enriched population. And we did TACMAN qPCR on those samples, total RNA. And these recapitulated what we saw in the whole mammary tissue. And I'll just talk about 3 here. You can listen to the data if you're interested. But this, again, genotype was significant. We're comparing the Copenhagen homozygous congenics here to the Wissenfurth strain, which is susceptible. And we can see lower expression in the resistant strain versus the susceptible strain for MIR3. And that's with or without giving DMBA. Okay, so then the next move was to see if we could track differences in MIR3 expression during mammary gland development. So Emily did this work using RNA-scope, and I've got representative images here where you can see during the embryo, the red puncta would indicate MIR3 presence, transcript presence, and then you can see at about 8 weeks of age, this is the age where we give the carcinogen, and then the adult mammary gland, virgin mammary gland shown here. And this is a graph of quantification. I didn't show you all the images, but highest expression is during embryonic development, and then it decreases and gets pretty low during adulthood, and then comes back a little bit during lactation, but then goes back down to a really low baseline. And she also saw most of her puncta in mammary ductal regions of the mammary gland, and then some in lymph nodes, and it was pretty low in stroma or adipose tissue, and in blood vessels, so endothelial cells. We also wanted to see what was going on during mammary carcinogenesis progression in the rat. So this is non-diseased rat mammary gland tissue here, and you can see ductal structure here, showing pretty low puncta for MIR3 probes. And then if you look in an invasive ductal carcinoma, you see quite a bit. So this would be similar to what is being seen in the human. And this increases in a disease stage already at atypical ductal hyperplasia. So this is hyperplasia that would be considered normal in the gland. You don't get a difference in MIR3 stain. And then MIR3 does increase in DMBA exposed rats as well. All right, so the big test now then, we had pretty much honed in on MIR3 being our candidate, best candidate for a causal gene. So we wanted to really knock it out and see what the effect on the susceptibility phenotype would be. So we collaborated with Kathy Krentz and Dustin Rubenstein at the University of Wisconsin. And what Kathy did is she used electroporation to put CRISPR-Cas9 system into Sprague Dawley embryos, and she ended up with 15 live pups. And Dustin designed those guide RNAs, and then his work resulted in 24 total mutations in 13 pups that did have edits. Over here in this table, you can see a list of the mutations.
David, sorry, could interrupt you. Sorry, because we're running out of time. Would you be able to wrap up shortly, please? That'd be great.
Sure. Yeah. Okay.
Thanks.
So what we did with those is we selected the mutations with premature stop codons, and you can see the effect on the amino acid sequence here. And those four are in the process or have been, founders have been bred to the N4 generation, and then brother-sister mating at N4 to do phenotyping work. And this is a result of that phenotyping. We can see the MIR3 targeted mutation rats have a much lower tumor incidence or tumor multiplicity than their litter mates who don't inherit any of the mutant alleles. So we've recapitulated the 50% reduction that we see in the MCS1B locus. And then there's also an effect on body mass where the homozygous mutant rats have lower body mass than their litter mates that don't have it, but we don't see an effect in the heterozygote. As far as starting to try to understand the mechanism here, so this is showing H&E staining of the targeted mutation mammary glands compared to the litter mates who have the wild type alleles, so non-mutant, and you can see normal eosin staining here, where in mutant targeted mutation mammary glands we see much less eosin, and this is all quantitated over here. So eosin stains protein non-specifically in the mammary gland. So collagen is the most abundant protein in the mammary gland, but there's also all kinds of extracellular matrix components in addition to that. So we're thinking that the mechanism might be through the extracellular matrix. So sorry about the technical difficulties, but hopefully I've convinced you that MIR3 is a causal breast cancer susceptibility gene, and that comes from a load of human genetics and rat genomics with expression data. And then the MIR3 editing, I think, really confirms it. And we do not know the role of the mammary gland or carcinoma development, so that will be next, but we think it might be through the extracellular matrix. Okay, so thank you. Probably have to take questions in the chat room then.
Apurva Chitre from, again, UCSD. Apurva. I'm not sure I pronounce your name properly.
No, you got it right.
All right, I'll give it a try. Thanks.
Right. So hello, everyone. I am Apurva Chitre, and I'm from the Palmer Lab at the University of California, San Diego. And today I will be talking about the genetic analysis of multiple measures of locomotor activity. And so the rats that we've used for this study are part of a large multi-site project, which focused on genetic analysis of behavior phenotypes which are related to drug abuse. And so the heterogeneous stock rats were sent to 3 different phenotyping centers throughout the US for phenotyping, and the rats were exposed to a different battery of behavior testing at each of these sites. And the Palmer Lab was involved in genotyping and performing genetic analysis of those behavior traits. So then, as part of this big project, locomotor traits were also measured in these phenotyping centers in different rats. And so I wanted to point here that the Tennessee cohort included adolescent rats. And so these rats were much younger than the Michigan and the New York cohorts. And so the combined sample size for these 3 phenotyping centers is approximately 4,200 male and female heterogeneous stock rats. And I also wanted to point out that these cohorts differed in procedural details, like the size of arena. For the Tennessee Center, the rats were placed in a much larger open field chamber as compared to the Michigan and New York cohorts. And the Michigan and the Tennessee cohorts also used an automated video analysis software whereas the New York cohort used beam breaks to track the behavior of the rats. And the duration of the test is also much longer for the Tennessee cohort as compared to the Michigan and New York. So there were procedural differences between the different cohorts. So we genotyped greater than 6 million SNPs per individual using the genotyping by sequencing method. And we estimated the genetic correlations, SNP heterogeneity estimates using GCTA. And we also used GCTA for mapping. And we used permutation-derived significance threshold. So one of the questions that we've addressed is, are the locomotor traits heritable? And the answer is yes. These do exhibit high SNP heritability, and these range from 0.2 to 0.3. Another question that we can ask is the genetic underpinning of these locomotor traits measured in different phenotyping centers, is it similar or is it different? And so what we've estimated is that the Tennessee Center showed the lowest genetic correlations with Michigan and New York, whereas the genetic correlation between the Michigan and New York locomotor traits was pretty high. It was 0.6. And so we think that one possible explanation for this low genetic correlation for the Tennessee locomotor trait could be that because of the open field locomotor activity in Tennessee includes a measure of anxiety, while the locomotor activity measured in a small box for the Michigan and New York cohorts, it does not include this component. Another explanation is that in Tennessee, the animals are adolescent, and we think that maybe during this developmental stage, the mechanisms underlying locomotor activity could be different. So we've also mapped these 3 locomotor traits. And so this is a combined Manhattan plot for the GWAS data for these 3 locomotor traits. And you can see that we've identified a total of 6 independent QTLs. And so these are colored by the top SNP for each of the QTLs for the 3 traits. Now let's zoom in into one of the QTLs. And so this one is on chromosome 2 for the Michigan locomotor trait. And so now we have zoomed in to just a few megabase on chromosome 2. And on y-axis, we can see the significance of the association. And we can also see the genes which are present in this QTL interval. And the focal dot is the top SNP for this QTL, the one with the most significant association. And the color of the dots represent the LDR square of the top SNP with the other neighboring SNPs. And so the red color indicates that these SNPs are in high LD with the top SNP. And so in order to determine the QTL intervals, what we've done is we have identified those markers which are in high LD with the top SNP. And so we've used a cutoff of R squared greater than or equal to 0.6. And so on the basis of this criteria, the interval size that we've determined for this QTL is 1 megabase. And in this 1 megabase interval, there are 2 genes. One of them is an uncharacterized gene, LOC gene, and the other one is this catenin delta 2 gene. And so the catenin delta 2 gene provides instructions for making the delta catenin protein. And this protein is active in the nervous system, where it helps in cell adhesion and it plays a role in cell movement. And in humans, this particular gene has been associated with autism and ADHD. So we think that this could be a potential candidate gene, which could be driving this association at this particular interval. All right. So before we talk about the next QTL, I wanted to talk about this RNA-seq data that we have, which was measured in 5 brain regions. And Daniel Munro from our group, who also gave a talk yesterday about this data set, has worked on calling eQTLs or expression QTLs for this particular data set. So now let's zoom into this chromosome 18 QTL for the New York locomotor trait. And so there are 10 genes in this interval. And the question we can ask is, are there any eQTLs that co-localize with the behavior QTL? And so for this same chromosome 18 locus, we can see that there are significant eQTLs, and it is associated with regulation of these genes in brain tissues that might be relevant for this locomotor trait. And so we think that this could be one of the things that might cause this locus to be associated with the locomotor behavior. And so if you notice, we've also calculated the LDR square of the eQTL top SNP with the GR top SNP. As you can see, for this particular gene, the R square is pretty high, it's 0.9. So we think that this could be one of the genes that may be causally related. So another question we can ask is, what other traits also show association at this same locus? And so this analysis is part of the CWAS pheno-wide association analysis. And so what we've done is we've looked at traits that also co-map in this 3 megabase window. And so we can see that there are some traits that co-map to the same region, and the peak SNP for these traits are in high LD with each other. Okay, so it's plausible to think that they are causally related to one another. And so the traits that I have highlighted here are from the same behavior paradigm, locomotor testing, and they are different measures of locomotor, which are activity and rearing. But the interesting one that comes up is the nicotine self-administration trait. And so from this, it shows that the locomotor behavior is associated with a more complex drug self-administration trait. And so this might make us think if there is a relationship between locomotor behavior and drug self-administration. And this kind of association might help us to interpret the meaning of the traits that we are looking at. And it will also help us to design follow-up studies about it. So we think that there is something about this particular locus that is influencing complex behavior traits. So in conclusion, we identified a total of 6 independent QTLs for 3 different locomotor-related traits. And we observed strong genetic correlations for locomotor traits between the Michigan and New York cohorts, but not for Tennessee. And the Tennessee Center, which showed the lowest correlation, it included younger animals, it used a larger open field, and it also included a longer test than Michigan and New York. And so now we are also reprocessing the data from the Tennessee cohort to focus on a shorter time period. And we are also analyzing a much larger data set. And so we think that with increased power, we will see if some of these loci are sex-specific. So I'd like to thank all the PIs and everyone involved in this work from the P50 NIDA Center. And thank you so much for listening.
Thank you, Apurva, for this fascinating talk. Thanks very much. Are there any questions for Apurva? I do have one, actually, if I may, just on the first one. So you showed that you gave some reasons why the Tennessee population deviates from the others.
Yeah.
I wonder if you've got enough data to actually run a linear model or so, and to see whether they actually come out as significant predictors. You could run some form of statistical analysis to verify that.
Yeah, that's actually an interesting point, and I think we will plan on running this as we have more data.
Thanks. Any other questions for Apurva? Don't see any hands.
Apurva, we have identified also catenin delta 2 for anxiety in the Eurotrans project. Yeah. We really need to talk more.
Yes.
Okay, great. Thanks very much, Apurva. So then we're moving on to our next speaker, Alexandria Salangi from Wake Forest University. Alexandria, over to you.
Hi, can you see my screen okay?
Yes, we can, yeah.
Okay, share that. I believe a new laptop. There we go. Okay. Pointer. All right, awesome. Hi, everyone. My name is Alexandria Salangi, and my talk today is on keratinocyte-associated protein 3, which we believe may impact eating behavior and adiposity differentially based on environmental influences. Obesity around the globe has been a major concern for a few decades now. So you can see the change from 1976 to 2016 for adults who are considered overweight or obese, to where you go from not a whole lot going on to a lot of dark blue on the map, which is a little concerning given all of the comorbidities associated with obesity. And so for most patients, they suffer from polygenic obesity, which is where they might have multiple genes that individually have small effects. But when you combine all of that together, you can have a much higher increased risk for developing obesity given certain environmental influences. So it's important that we better understand the genetic architecture of obesity to develop better treatments and preventative measures to prevent the disease. Now, our lab primarily uses the HS rat, the heterogeneous stock rat, which has been talked about before, so I won't go into too much depth on it. But the genetically diverse outbred rat model allows us to genetic fine map complex traits, such as those different adiposity traits, including retroperitoneal fat mass, which is a visceral white adipose depot that surrounds your kidneys and is sort of on the back of both rats and humans. A previous study from our lab actually found a retrofat QTL on rat chromosome 6 in HS rats. And you can see the signal here. And what was really interesting is when we looked at the haplotype data for that QTL, is we found that those HS rats with the inbred wiki haplotype at this particular region of chromosome 6 had decreased retrofat mass compared to the other HS rats. We also looked at expression of different genes based on RNA-seq in these HS livers, and we found a co-localizing cis-EQTL on rat chromosome 6. And when we look at the haplotype data for expression of CRITCAP3, we found that those rats with the wiki haplotype not only had decreased retro fat mass, they also had increased expression of CRITCAP3. And when we condition the retrofat QTL scan based on CRITCAP3 expression, you can see that the signal completely disappears, demonstrating that CRITCAP3 is a full mediator for this retrofat QTL, and expression of the gene is potentially explaining the phenotype. So it's potentially worth looking at. What we also saw in the HS rats is that as CRITCAP3 expression increases in these rats, we saw a correlation where retro fat mass is decreasing, again suggesting a relationship worth looking into. And for what's also important is that we don't necessarily care about obesity in rats. We want to have a human connection. And we see that CRITCAP3 actually falls within a locus that approaches significance for human waist to hip ratio, which is one of the measures for humans for obesity risk. And so that's demonstrating that CRITCAP3 is a gene worth looking into. And it's a novel gene for adiposity that no one else has really talked about in previous papers. So our first step was to create a CRITCAP3 knockout rat. And we used CRISPR-Cas9 to delete 2 base pairs from the gene. And we worked on the inbred wiki background strain because that was the strain that had increased CRITCAP3 expression. And this work was done by the Gertz lab at the Medical College of Wisconsin. And our hypothesis here was that as CRITCAP3 expression is decreased in our knockout rats, we should see a corresponding increase in adiposity in these rats. We successfully demonstrated that in our knockout rats, there was decreased CRITCAP3 expression, which is important. And we also saw at the 6 weeks of age mark, increased body weight in our knockout rats, both for males and females, supporting our hypothesis that our knockouts will have increased adiposity. A very brief methodology of the 2 studies that I'll be talking about today is that diet starts for our rats at 6 weeks of age, and that's our 0 weeks on diet marker. And they continue diet for 13 weeks, at which point they are euthanized and their tissues are dissected. And for both studies, we looked at males and females on a high-fat diet and low-fat diet. But for the sake of time, I'll really only be focusing on our high-fat diet females, where we saw the largest adiposity effect. In study 1, we saw that our knockout females were gaining more weight compared to their wild-type siblings, as you can see on the growth curve here. They were also consuming more food over the course of the study. And at the end of the study, when we looked at retro fat mass, you can see that the knockout females on a high-fat diet and low-fat diet had increased adiposity compared to the wild-type groups. So all of this was supporting our original hypothesis that knockouts with decreased CRIT-Cap3 expression had increased adiposity, supporting CRIT-Cap3 as a gene for adiposity. We sought to replicate this study, and unfortunately, we failed to replicate that really beautiful adiposity phenotype. So you can see in the growth curve here that our wild-type females are a little bit heavier than our knockout females over the course of the study, although they aren't really gaining that much more weight. There's no differences in food intake over the course of the study between wild-type and knockout rats. And at the end of the study, when we look at retro fat mass, we aren't seeing any significant differences between wild-type and knockout females. So we failed to replicate the adiposity phenotype that we had been expecting based on our original data and our beautiful study 1. What I wanted to do was look at, okay, which group changed between these 2 studies, because we went from a really nice phenotype to no phenotype. And what I found is in comparing the 2 genotypes across the 2 studies, is you can see a significant increase in the fat mass of our wild type group without any corresponding increase in our knockout group. So for some reason, our wild type rats were getting fatter with no change in our knockouts. And this is also supported if I look a little bit closer at the growth curve and food intake of just the wild type rats between the 2 studies. You can see that in study 2, the wild type rats are gaining a lot more weight compared to what they were before. And with that, they're also eating a lot more compared to what they were before, with no corresponding changes in our knockout rats. The first thing we tried to look at were there any sort of environmental changes that maybe affected their behavior? There was no differences in temperature between the 2 studies, no differences in humidity in the room. We used the same diet from the same source for both studies, and there were no major changes in personnel between study 1 and study 2 on our end in the lab or in the animal resource program more regularly taking care of the rats. What we do know caused an environmental change was COVID. So prior to COVID in study 1, our building was pretty noisy and the surrounding area was pretty noisy. So we had construction on a nearby apartment building. The space right next to our vivarium is actually used as an event space. So can be a lot of music, a lot of people, just a lot of noise in general. And there was also, of course, higher traffic in the vivarium because we had labs doing active research, as they normally do. Post-COVID shutdown, construction had finished, so there was no construction nearby. There were no events in the building because the building was closed, and a lot of labs actually paused their animal research. So there was also reduced traffic in the vivarium for study 2. So overall, a much quieter environment. What we know is that noise impacts body weight of rats. And so this is just demonstrating from a 2017 study that rats that had been exposed to construction noise had a decreased body weight compared to control rats that weren't exposed to that noise. And we saw similar changes in our own HS rats that were in the same room as my CRIT-Cap3 colony. And you can see that between pre-COVID and post-COVID, a significant increase in the body weight of our female HS rats, which led us to the hypothesis that there was a different environment between study 1 and study 2 that seemed to be affecting the body weight of all of the rats in the room, not just my colony. What we also know is that noise increases corticosterone levels in rodents, marker of stress. And so this study from 2013 is showing that for rats exposed to construction noise, significant increase in cort levels. We actually sent out to the Rade lab at Northwestern University serum from our study 1 females at SAC to look at their cort levels. And what we found that was really interesting was that between our knockout and our wild type groups, our knockouts had really low levels of cort, and our wild types had comparatively higher levels of cort, potentially indicating increased stress. So this led us to the hypothesis that if our environment had been different, wild type and knockout seemed to be responding to that environment differently in study 1. So that led us to the idea that maybe CRIC-CAP3 is acting in a stress pathway. And that's the question that we really want to answer moving forward. So we're going to focus on our high-fat diet females where we had that beautiful adiposity phenotype, the first study. And so we'll have a group of no stress rats and a group of stressed rats. And if our hypothesis is correct, we are expecting that our no stress control group will essentially mimic study 2, where there's no differences in adiposity between our wild type and knockout group. And our stressed group will mimic study 1, where our knockouts should have increased adiposity because our wild types won't be eating as much. So with that, I would like to acknowledge and thank everyone in my lab. I would like to acknowledge and give a shout out to everyone in the Gertz lab and the Rade lab who have contributed to the project and thank them for their help. Acknowledge the 2 funding sources for the project and for me. And if you have any questions or feedback, please feel free to send me an email. I'm happy to talk. With that, I'd like to thank you for your time and please let me know what questions you have. Thank you.
Super. That's a really fascinating talk, Alexandria. I really enjoyed that in particular because we had a similar episode in our animal unit pre and post COVID, and it turns out it wasn't just the staff, the routine had changed, but also that the light sensors were faulty, and that caused increased stress levels in one particular section of the room, and it affected development, body weight growth, etc., and reproduction. It's a total mess, to be honest, but it took us a while to figure out what was going on. Just wondering about the diet as well, because that happened to us as well, in that the calorific value of the diet that came from the same supplier showed a big batch effect, basically. I don't know whether you look at this at all, whether you just trust the suppliers. I stopped trusting suppliers of mice and food now because we've had so many bad experiences, but that could also be another reason for that. But fascinating. Anyway, any comments and other questions for Alexandria?
I've got a question. So Alex, have you guys noticed any change? And actually, this could be for anybody. Any behavior changes as far as breeding ability or litter size or?
Yeah, so this actually all came about from a conversation I was having with a friend where for her lab, they had noticed a change in breeding from actually when they moved to Wake Forest at our biotech place where the noise caused a decrease in breeding there. For my colony specifically, I haven't noticed any major changes pre and post COVID, but the wiki rat isn't always a great breeder to begin with. And our HS rat breeding colony is actually at a separate facility now that doesn't get exposed to all that noise level. So no changes on my end, at least.
Hi, Alexandria. I have a quick question here, if you don't mind. So often chronic stress can be associated with sort of reductions in lean mass and then also hyperglycemia or insulin resistance. Did you notice that in different between your groups?
We didn't see any changes in lean mass in the females, but I didn't bring it up here, but we actually performed IPGTT on a group of the study 1 females, and then we had to stop because of COVID for the last few cohorts. But our wild type animals actually had sort of worsened not significantly, but worsened glucose tolerance than our knockouts, which was surprising given the differences in body weight between them. Any other questions? All right, I will stop sharing. Feel free to send me questions in the chat or shoot me an email. Thank you all for your time.
Thanks very much, Alexandria. Again, that really fascinating talk. Thank you. All right, in which case we can move on to our next speaker, Lauren Kuffler from the Jackson Laboratory. Lauren.
Present our work. My name is Gautam, and this is a joint work with Keith Shepard and Vivek Kumar. This is the title of my talk. So gait and posture are sensitive measures of proper functioning of numerous neural circuits. This is because proper gait, balance, and posture are under the control of multiple nervous system processes. The ability to quantitate gait and posture accurately at high precision in humans has shown that these are perturbed in numerous conditions. Many illnesses such as Down syndrome, autism spectrum disorder, schizophrenia are affected by, they lead to poor gait and posture deficits. So from a clinical perspective, automated high throughput and sensitive gait and posture analysis has been a powerful tool to detect disease states in humans. So so modern animal gait analysis methods are credited to Hildebrand, who classified gait based on quantified matrix. So he defined a gait cycle in terms of contact of limbs of the animals to the ground. This is in contrast to human gait and posture analysis, which is akin to quantification of whole body movement rather than just contact of limbs with the ground. So the ability to combine open field measures with gait and posture analysis would offer key insights into neural and genetic regulation of animal behavior. So new technology has enabled a new era of animal behavior analysis from video-based open field assays. We leverage modern neural network methods to carry out mouse gait and posture analysis in the open field in our recent work. We developed a system that can measure gait and posture from a top-down perspective that is invariant to high level of visual and morphometric diversity in mice. It offers very high precision and accuracy and was validated with known gait mutants, several mouse models of autism spectrum disorder, and mice from 62 isogenic mouse strains. We described that in this paper. Now we would like to apply our methods to JAX-COM2 pipeline and provide statistical evidence that gait and posture are often perturbed in single gene deletions. So here's a pictorial description of a pipeline. We have 8 cameras which record freely behaving mice in open field. We do this by placing overhead cameras in the COM field arenas. We use a deep convolutional neural network to perform pose estimation of mice on top-down video of an open field. This network provides 12 key points for each frame of the video describing the pose of the mouse. The 12 key points are the nose, the left and right ears, the base of neck, the left and right forepaws, mid-spine, the left and right hindpaws, the base of tail, mid, and the tip of the tail. So we have developed downstream components that are capable of processing time series of processes and identifying intervals that represent individual strides. So we can extract several important gait metrics on per stride basis. Here's an illustration showing how we derive 3 spatial stride measurements from hind paw foot strike positions. As mentioned earlier, we're also interested in looking at whole body posture movement, which is measured by quantifying the movement of tip of nose, the base of the tail, and the tip of the tail. And so we have certain linear measures which measure the gait, and then there are some circular measures which measure the posture of the mouse, how the mouse moves. And these metrics provide a consistent view of the structure of mouse gait and whole body posture. So we analyzed 6 months of data, which consists of 2019 mice from 240 mutant lines. We asked the question, can we capture differences in gait across different mutant lines using our methods? And we carry out two separate analyses. We carry out analysis at a mutant line level and at an animal level. In the analysis at the mutant line level, we require that our mutant lines have at least 5 animals tested. So there are 162 mutant lines with at least 5 animals tested. And in the per animal analysis, we include all mutant lines. So even the lines which have less than 5 animals tested. This is the line which is our control, the black 6NJs, which has 189 animals tested in that line. So we had to do some data pre-processing. So we focus on our analysis on only a certain speed bins, and only look at some bins with certain angular velocity. So we do this to make sure we're analyzing mice walking in approximately straight direction and at a similar speed. We also require animals to have at least 75 strides. And we plotted the phenotypes that we measured over time to rule out any systemic changes that may be due to fluctuations over time in the data produced by the COM pipeline. So we don't see any fluctuations. It's quite stable. Our phenotypes, here's an example of step width. So we asked the question, does a given gene deletion affect the linear gait and posture phenotypes? So we use a linear mixed modeling framework to analyze these phenotypes. Our models adjust for temporal variation arising due to multiple batches. And these batches have been tested over 6 months. And we looked at 3 models. So M1 tests looks at adjusts only for body size. So it can reveal mutant lines that have changes in stride speed. We looked at a model which adjusts for changes in stride speed and detects strains that have changes in stride due to body size differences. However, we found that this model M3, which adjusts for both body size and speed, identifies strains with altered gait. With altered gait after adjusting for body sizes and stride speed differences. So in the left block here, on the left here in the left block, the number of stars represents the strength of evidence against the null hypothesis, which is that there is no genotype-based effect. The pluses represent a suggestive difference. And on the right, we have the effect sizes, the standardized effect sizes, and the color red for positive and blue for negative represent the effect sizes, direction, and magnitude. So using these three different models, we found different number of mutant lines having significantly different gait defects when compared with controls. Next, we looked at the phase differences, the body postures, and these are circular in nature, so we require certain different types of analysis methods. So we use a circular linear regression model that adjusts for body size and stride speed of the animals, and we found 5, 28, and 21 mutant lines to have significantly different phase differences identified by these models. And here's like a vignette of what these phase differences look like across different lines and mutants, across different lines. These analyses were all univariate. We wanted to look at the multivariate analysis because univariate analysis assumes that the gait phenotypes are independent and when they may not be independent. So multivariate analysis, we asked the question of, are there any mutant lines that are outliers? So we used the PCA-based algorithm, and it revealed to us some certain outliers, which are shown in red here. So it revealed certain lines which are not previously identified as having significantly different gait defects compared to the controls. We have 9 gait phenotypes. We wanted to see if there exists a lower dimensional gait space where the mutants separate from the controls. So we used a linear discriminant analysis to quantitatively distinguish between mutant lines and controls. So for example, here individual dots represent the animals the colors represent different mutant lines. And we see that the x-axis, the LD1 axis, effectively separates the controls from the mutants, and the LD2 axis separates these mutants from these mutants. If you look at the contribution of different gait phenotypes to each of these axes, you'll see that they have different contributions, like step width contributes less than speed to this axis and helps in separating the controls from the mutants. So until now, we have looked at only mutant lines where at least 5 animals were tested. So we also wanted to identify mice that are outliers due to genetic reasons, which were missed earlier because of the underpowered study. So less than 5 animals were tested in that line, and we couldn't include that. Or we couldn't identify, or they were outliers due to non-genetic reasons like injury or developmental disorders. So we used a outlier detection technique. So lines not analyzed previously included 52 outlier animals. Here are the outlier animals that show up. We also were able to detect 2 previously identified animals in one of our previous works that showed abnormal movement in the open field. When we looked at the videos of certain animals, we found these animals to have injuries, they were limping, and so we were able, the analysis at the animal level helped us identify animals with idiosyncratic differences, which emphasize the sensitivity of our approach in identifying these animals. We performed an analysis at 2 levels, at the strain and at the animal level. For univariate analysis, we used the linear mixed modeling approach. For the multivariate, we used the LDA approach. So all these analyses combined, we found 61 strains with gait defects out of the 240 strains. And we compared these findings with the CSD, the Sherpa dysmorphology test assay that is conducted at JAX, where around these many mutant lines are tested, and less than 0.001% of them are reported to have gait defects. So these are these lines that were reported by the JAX assay. And both our univariate and multivariate approaches reported the ARPC5L strain as having a gait defect. Interestingly, we also found animals in strains RAB6, RAB6B, and LAM5 as outliers in our analysis at the animal level. So to conclude, I mean, our high throughput analysis reported strain lines with gait deficits found by human observers, and also identified lines missed, thereby emphasizing our approach's utility and sensitivity. So our framework consists of using 12 key points to detect the anatomical parts of the animal with several downstream components, and extract several classical linear measures of gait and the whole body posture matrix. We used standard statistical modeling approaches to find phenodeviant individuals for mean and variance phenotypes. And we used multivariate tools to identify mutant strains as well. We show that when we compare our results from our framework with those obtained by a trained human expert, and we see that our methods are much more sensitive than human observers. Thank you.
That's brilliant. Thanks very much, Gautam, for that. You've got a lot of questions in the chat. You might want to go through them if you want to use your break that's coming up in a minute. But I'm just going to pause here and see if anyone's got any questions they want to ask right now.